how can organisms synthesize complex molecules like proteins, carbohydrates, lipids, and nucleic acids? These biological molecules are generally known as polymers. And a polymer consists of multiple subunits called monomers. So if we put a whole bunch of one type of monomer together, we get a polymer. But how do we put them together? Biological polymers are created by a process called a condensation reaction. This process can also be called dehydration synthesis and creates chemical bonds between the monomers. Now, the reaction in reverse can break those chemical bonds, and this process is called hydrolysis. The names may not make much sense right now, but let's see how these processes actually work. To join two biological monomers, a hydrogen and hydroxyl group are removed, and water is formed. The shortened ends of the monomers connect in a covalent bond. Condensation is a term that basically means formation of a liquid. Since a condensation reaction results in the formation of a bond due to the removal of two hydrogens and an oxygen, this is an appropriate term. Hydrolysis is just the opposite. If a polymer needs to be split up, water can be added to break the covalent bonds between the monomers. Hydrolysis literally means water splitting. Sugar is a general term to use for carbohydrates, but be aware all sugars are not sweet. This is a picture from a microscope image, called a micrograph, from a cross-section of a plant root. Two carbohydrates are stained, cellulose and starch. Cellulose is stained green and is found in the cell walls of the plant cells. Starch is stained purple and is found within the cells. Both cellulose and starch are complex carbohydrates, and we'll learn more about them later on. All carbohydrates have a very similar structure. In most carbohydrate molecules, you will find two hydrogens for every carbon and oxygen atom. The N subscript means that we can have different multiples of this arrangement. For example, if we input a 5 for N, we will get a molecule with 5 carbon, 10 hydrogen, and 5 oxygen atoms. As in most organic molecules, the backbone is composed of carbon atoms. Attached to the carbon skeleton are hydroxyl groups that allow most carbohydrates to readily dissolve in water. This is how each monomer, specifically called a monosaccharide, of a carbohydrate is composed. Carbohydrates may consist of one or many monosaccharides bound together. Carbohydrates serve many functions in living organisms. Structural carbohydrates like cellulose and chitin provide strength and stability. When carbohydrates are bound to proteins, they form glycoproteins, which serve as identification markers on cells. And carbohydrates such as starch and even glucose store energy that can later be used by the organism. We will discuss some of these functions in more detail later on. In addition to hydroxyl, we also find carbonyl functional groups in sugars. If the oxygen is double bonded to a carbon at the end of the skeleton, we call this specific type of carbonyl an aldose. But if the carbonyl occurs in the middle of the skeleton, the carbonyl is referred to as a ketose. Most monosaccharides, the monomers of carbohydrates, have 3, 5, or 6 carbons. If the monosaccharide has 3 carbons, it is called a triose. Glyceraldehyde is a triose that is produced during photosynthesis. This monosaccharide is used to synthesize more complex monosaccharides. A 5-carbon sugar is called a pentose. Deoxyribose is a pentose found in DNA. A 6-carbon sugar is called a hexose. And while glucose is the monosaccharide used by most living organisms, organisms utilize many different hexoses for various reasons. Now, as you learned at the beginning, carbohydrates are composed of a ratio of carbon, oxygen, and hydrogen atoms. So if hexoses have 6 carbons, how many hydrogen and oxygen atoms would they have? Yes, 12 hydrogen and 6 oxygen. But even though most hexoses have this molecular formula, the atoms are arranged in different ways. Molecules that have the same molecular formula but different structures are called isomers. For example, glucose has the molecular formula of C6H12O6, with a hydroxyl group attached to the 4th carbon in the same position as the hydroxyl on the 2nd carbon, while galactose has the same exact molecular formula. C6H12O6, but the hydroxyl group is in a different position. This may not seem like a big difference, but the position of the hydroxyls give glucose and galactose slightly different properties. And these bonds found in carbohydrates are very important. Carbohydrates sustain life as they are the products of photosynthesis, storing solar energy in the form of chemical bonds. They are ideal for storing usable energy due to the high percentage of shared electrons in the molecule. All those carbon and hydrogen atoms bonded together have similar electronegativities. Therefore, the electrons are not bound as tightly to the atoms. When the bonds are broken, there is more free energy to power endergonic reactions. But we did mention that glucose is the most important monosaccharide in organisms, so let's look at it in a little more detail. In aqueous solutions, such as the solutions found inside cells and the extracellular fluid, glucose does not typically exist in a linear state. As you can see, the carbons are numbered, from the terminal carbon on one end of the linear molecule, carbon 1, to the last carbon in the backbone, carbon 6. And in aqueous solutions, the hydroxyl attached to the fifth carbon and the carbonyl interact to form a ring structure. The formation of the new bond displaces the hydrogen of the hydroxyl. In addition, a hydrogen atom forms a bond with the oxygen of the carbonyl, converting it into a hydroxyl functional group. But we also have to consider the position of that hydroxyl group on the first carbon. The ring can form with the hydroxyl group in this position, and we would call the molecule alpha glucose. Or in this position, and we would call it beta glucose. What would we call these two different forms of glucose? Yes, isomers, because they have the same molecular formula, but different structural formulas. So, if monosaccharides are the monomers of carbohydrates, how are they added together to form polymers? By a condensation reaction, of course. In carbohydrates, the covalent bond that forms between the monosaccharides is called a glycosidic linkage. Let's look at what we can form when two monosaccharides are joined. Disaccharides are carbohydrates composed of two monosaccharides joined by a glycosidic linkage. Some common disaccharides include sucrose, common table sugar, composed of the monosaccharides glucose and fructose, lactose, the sugar found in milk, composed of glucose and galactose, and maltose, a sugar derived from barley for fermentation to produce drinking alcohol, composed of two glucose molecules. So here's two glucose monomers. As you can see, they are both alpha glucose since the hydroxyls on the first carbons are in the down position. In addition, the sixth carbon is in the up position. To form a glycosidic linkage, a condensation reaction must occur. And, of course, it is the removal of a water molecule that forms the linkage. In this case, we have formed a very specific linkage. Since the two glucose molecules were the alpha forms, we call the covalent bond that has formed between the monomers an alpha 1,4 glycosidic linkage. The 1 and 4 refer to the fact that the bond occurs between the first carbon of one monosaccharide and the fourth carbon of the other monosaccharide. The resulting disaccharide is maltose. And if you will notice, the sixth carbons of the monomers are still in the up position after the bond is formed. Now let's take two different monosaccharides, galactose and glucose. This glucose is different, as it is the beta form, with the hydroxyl on the first carbon in the up position. Take a close look at the beta galactose. How does it differ from the beta glucose? Right, they have the same molecular formula, but the hydroxyls on the fourth carbon are in different positions, and that makes them isomers, right? And when the condensation reaction occurs, a beta 1,4 glycosidic linkage is formed, giving us the disaccharide lactose. If you will notice, the sixth carbons do not remain in the same positions after the linkage forms. The glucose molecule must flip upside down, so that the sixth carbons are in the opposite positions in this disaccharide. Now, do we have to stop at 2 monosaccharides? No. In fact, very long chains of monosaccharides can form to produce polysaccharides. These chains may have hundreds or thousands of monomers. Common polysaccharides include starch, glycogen, cellulose, chitin, and peptidoglycan. Let's look at each of these in more detail. Starch is the polysaccharide plants form in order to store their glucose for later use. The potato plant stores its excess starch in the potato. If we look at the cells in a potato, we see the small structures amyloplast that contain the starch. And if we look at the starch, we can see it is composed of 2 forms, helical amylose and branched amylopectin. The glucose monomers in amylose are joined by only alpha-1,4-glycosidic linkages, while the amylopectin has alpha-1,6 glycosidic linkages at the branch points. Glycogen is the polysaccharide animals form in order to store their glucose for later use. Glycogen is commonly stored in the muscles and liver, specifically in granules throughout the cell. Glycogen consists of glucose joined by alpha-1,4-glycosidic linkages. Its branch structure is similar to amylopectin, but glycogen does contain more alpha-1,6 linkages. Cellulose is the polysaccharide plants form for structural purposes. If we look at a plant, its cells are surrounded by a tough cell wall. A major component of cell walls is cellulose, which consists of glucose joined by beta-1,4-glycosidic linkages. These long, straight chains are further joined by hydrogen bonds to make cellulose a very strong, stable building material. While cellulose is a great structural material in plants, it is a bit of a problem for those who eat plants. Most organisms cannot digest cellulose. This is because they lack the enzyme to hydrolyze the beta-1,4-glycosidic linkages, making the glucose unattainable. In fact, only certain bacteria can break down cellulose. Organisms who do eat large amounts of plant material, such as ruminants, like cows, or termites, house cellulose-digesting bacteria in their guts. But cellulose is also a main part of the indigestible polysaccharides considered to be fiber, which keeps the digestive tracts of humans functioning normally. Chitin is the polysaccharide fungi and other organisms such as insects use for structural purposes. Chitin is not composed of glucose molecules. Instead, the monosaccharide found in chitin is called N-acetylglucosamine, represented by the blue molecules in the above diagram. It is similar to glucose, but one of the functional groups contains nitrogen. The monomers are joined by beta-1,4-glycosidic linkages. Hydrogen bonds form between the monomers of the chains, increasing the durability and strength of this polysaccharide. Peptidoglycan is the polysaccharide bacteria use for structural purposes. Like chitin, peptidoglycan is composed of N-acetylglucosamine monomers. But it also contains another type of monosaccharide. These are joined by beta-1,4-glycosidic linkages, and the peptidoglycan chains are linked together by short strands of amino acids.